Hello students, today I bring for you a new chapter, Cell, the Unit of Life. It's a class 11th chapter biology. This chapter is fully resemblance with class 9th, the fundamental unit of life, but some more things we are going to learn in this chapter comparing to class 9th. So it is a easy one. I hope this video will helpful for your better preparation in upcoming exams. So let's start. Cell. A cell is the fundamental structural and functional unit of all living organisms. Introduction. Robert Hooke, we all know, who discovered the cell in 1665, and cord cell discovered by Hooke. Then Anton von Leeuwenhoek came and he observed a living cell in pond water. Next, the invention of compound and electron microscope reveals the structural details of the cell, and we all know about the microscopic organisms. Cell theory. Malthus Selden 1838 observed that plants are composed of cells, and Theodore Swan 1839 reported that the cell had a thin outer layer, plasma membrane. He also found that plant cells have cell wall. He proposed a hypothesis that animals and plants are composed of cells and product of the cell, and these two formulated the cell theory. Then Rudolf Virchow 1855 explained that cell divided and a new cell are formed from pre-existing cells in his own book that is called Omnis Cellulae e Cellula. He modified the cell theory, and according to R. Virchow, new cell theory will be all living organisms are composed of cells and products of cells, and all cells arise from pre-existing cells. An overview of cell, which having all cells contain cytoplasm, which is a semi-fluid matrix where cellular activities and chemical reactions occur. This keeps the cell in living state. And ribosomes. It is a non-membrane bound organelle found in cytoplasm, chloroplast, mitochondria, and on rough endoplasmic reticulum. Smallest cell, mycoplasma, 0.3 micrometer in length, which is also called PPLO. Okay, PPLO, pleuropneumatic like organism, PPLO. Largest isolated single cell, that is egg of an ostrich. Longest cell, nerve cell. Size of bacteria 3 to 5 micrometer. Human RBCs 7.0 micrometer in diameter. Here are some diagrams. Next, based on functions, shape of cell may be disc-like, polygonal, columnar, cuboid, thread-like or irregular. Here you can see the structure of the cell. Here a parenchymatous tissue, collenchyma tissue, sclerenchyma tissue, and in this side animal cells like nerve cell, muscle cell, bone cell, gland cells, blood cells, and reproductive cells. Cells are two types, prokaryotic and eukaryotic cell. You all know about what is a prokaryotic cell, what is a eukaryotic cell in previous classes. So let's start the prokaryotic cells. They have no membrane bound nucleus and organelle. They include bacteria, blue-green algae, mycoplasma and PPLO, pleuropneumonia like organisms. They are generally smaller and multiply more rapidly than eukaryotic cell. They vary in shape and size. As you can see, here are some bacteria, cyanobacteria and mycoplasma, PPLO. Some diagrams are there. By watching these diagrams, your doubt or the topic will be easily cleared properly. Next, bacteria having four basic shapes: bacillus or rod-like structure, coccus or spherical, vibrio or comma-shaped, spirillum or spiral. As you can see in the diagrams, all four basic shapes of bacteria are in the diagrams. Next is cell organelles. Prokaryotic cell organelles. What are they? Now cell envelope, mesosome and chromatophores, nucleoid, flagella, pili and fimbriae, ribosomes, inclusion bodies. First of all, cell envelope. It is a chemically complex protective covering. It is made up of three tightly bound layers: outer glycocalyx, middle cell wall and inner plasma membrane. Next, glycocalyx, it is the outer layer. It is composed of and thickness vary in different bacteria. It may be a slime layer, loose sheath or capsule, thick and tough. So in some bacteria, it may be in a loose sheath, that is called slime layer, the glycocalyx, or a capsule, thick and tough. Cell wall, it is the middle layer. Seen in all prokaryotes, it gives shape to the cell and provides a structural support to prevent the bacterium from bursting or collapsing. It is the middle layer cell wall. You can see that in the diagram. Next, third is plasma membrane. It is the inner layer. It is semi-permeable in nature and interact with the outside. This is structurally similar to that of the eukaryotes. So here the plasma membrane is similar to eukaryotes. Next, cell envelope. Based on type of cell envelope and response to Gram staining, bacteria are two types. What is Gram staining? Gram staining means coloring. If you add some bacteria in a particular stain or color, some bacteria absorb the color or some are not absorb the color. So it was developed by Christian Gram, and according to this, he giving the name Gram staining. So those bacteria that take up the particular color and become colorful, they are called Gram positive, and those do not retain any color. Either they added in a particular staining material in a particular color, they are called Gram negative. As you can see in this diagram, E. coli and Staphylococcus aureus. There are two different type of bacteria. When added in a particular color, the Staphylococcus aureus absorbs the color of the environment and becomes colorful, as you can see in the blue color. But E. coli does not absorb the color. So here the Staphylococcus is Gram positive, while the Escherichia coli is Gram negative. Mesosomes and chromatophores. These are membranous structures. Mesosomes, as you can see in the diagram, it is formed by the infolding of plasma membrane. As you can see labeled here, mesosome. It includes vesicles, tubules and lamellae. Functions of mesosome. For cell wall formation, mesosome helps in the formation of cell wall. For DNA replication, for distribution of chromosomes to daughter cell, for aspiration and secretion, to increase the surface area of plasma membrane and enzymatic content. So these are the functions of mesosomes. Next is chromatophore. These are pigment containing membranous enfoldings in some prokaryotes, example cyanobacteria. So chromatophores are not present in all the bacteria. It is found in some type of bacteria, and these are the pigment containing membranous foldings. Okay, mostly found in some prokaryotes, example cyanobacteria. Next is nucleoid. It is formed of non-membranous or naked circular genomic DNA, single chromosome genetic material and protein. Many bacteria have small circular DNA, that is called plasmid, outside the genomic DNA. It gives some unique phenotypic characters, example resistance to antibiotics to bacteria. As you can see in the diagram, nucleoid and plasmid is present outside of the genomic DNA. Flagella. These are thin filamentous extensions from the cell wall of motile bacteria. Their number and arrangements are varied in different bacteria. Bacteria flagellum has three parts: filament, hook and basal body. What's the diagram? The filament is the longest portion and extends from the cell surface to the outside. Filament is the longest portion. Next is pili and fimbriae. These are surface structures that have no role in motility. So pili and fimbriae, they have no role in motility, in movement. Pili, singular pillars, are elongated tubular structure made of a special protein that is called pillin. Fimbriae are small bristle-like fibers sprouting out of the cell, as you can see in the diagram. In some bacteria, they help to attach the bacteria to rock in streams and to the host tissue. So their work is the to attachment of the bacteria in a particular rock or a particular host tissue. Next is ribosome. They are associated with plasma membrane and of prokaryotes. Plasma membrane of the prokaryotes. They are about 15 nanometer by 20 nanometer in size. They are made up of two subunits. So the ribosome is made up of, prokaryotic ribosome is made up of two subunits, 50S and 30S unit. They together form 70S prokaryotic ribosome. Here you do not have to add 50 plus 30, 80. Here you just to remember there are two different subunits of the prokaryotic ribosome. One is 50S or larger subunit and other is 30S, that is the small subunit. Both forms together 70S prokaryotic ribosome. S stands for sedimentation coefficient, a measure of density and size. So here you can see 70S and 30S. This is the particular S for sedimentation coefficient. As you can see in this diagram, ribosome is present, as well as here you can see small subunits. 30S plus 50S gives rise to 70S. Here prokaryotic ribosome, 50S subunit, 30S subunit and forms 70S prokaryotic ribosome. In eukaryotic, 60S subunit plus 40S subunit and makes 80S. Okay, even in 60S subunit there are three different parts: 5S RNA, 5.8S and 28S RNA, and 40S subunit 18S RNA. We are going to cover this topic in further classes. Just remember, the prokaryotic ribosome is 70S, which is made up of two subunits, 50S and 30S. And eukaryotic ribosome is made up of 60S plus 40S, makes it 80S ribosome. Next, functions. Ribosomes are the site of translation, or you can say protein synthesis. Several ribosomes may attach to a single mRNA to form a chain called polyribosome or polysome. Ribosomes of a polysome translate the mRNA into proteins. So the main functions is to protein synthesis. Okay, as you can see, larger subunit, smaller subunit, they combine with the mRNA and they make the process translation, and here the peptide chain, polypeptide chain or protein is formed, and again in the end both the subunits are again divided. Okay, next they find another mRNA. mRNA means messenger RNA. Okay, M stand for messenger. Next is your inclusion bodies. These are non-membranous stored reserve material seen freely in the cytoplasm of prokaryotic cell. Inclusion bodies, as you can see in this diagram, it is labeled. Example: phosphate granules, cyanophycean granules and glycogen granules, gas vacuoles, etc. Gas vacuoles are found in blue, green and purple and green photosynthetic bacteria. Remember the points. Next is the eukaryotic cell. They have well organized membrane bound nucleus and organelles. Presence of membranes gives clear compartmentalization of cytoplasm. They have a complex locomotory and cytoskeletal structure. Their genetic material is organized into chromosomes. Organelles, cell organelles includes cell membranes, cell wall, endomembrane system which includes Golgi apparatus, lysosome and vacuoles, mitochondria, plastids, ribosomes, cytoskeleton, cilia and flagella, centrosome and centriole, nucleus, and your final microbodies. As you can see, this is the plant cell and this is the animal cell. You are familiar with this type of cell structure in previous or lower classes. First of all, cell membrane. Chemical studies on human RBC show that the cell membrane is composed of lipid bilayer, proteins and carbohydrates. As you can see in this diagram. It is the same diagram that is also in your NCERT. Lipid bilayer and proteins, sugar or carbohydrates. Lipid mainly phosphoglycerides have outer polar head and inner hydrophobic tail. The lipid layer have outer polar head and inner side is hydrophobic tail. So the non-polar tail of saturated hydrocarbons is protected from aqueous environment. Ratio of proteins and lipids varies in different cells. Example, in human RBC, membrane has 52% protein and 40% lipid. So this ratio is vary in different cells. Based on the ease of extraction, membrane proteins are two types: integral protein, which is partially or totally buried in the membrane. As you can see, integral protein. Peripheral proteins lies on the surface of the membrane. Okay. These two, channel protein, carrier protein, are integral proteins, while the peripheral protein you can easily identify in this diagram. Fluid mosaic model of cell membrane, proposed by Singer and Nicholson in 1972. According to this, the quasi-fluid nature of lipid enables lateral movement of proteins within the overall bilayer. So the quasi-fluidic nature of the lipids that helps and enables the movement of proteins within the overall bilayer. This ability to move within the membrane is measured as its fluidity. This is called fluid mosaic model, which is proposed by Singer and Nicholson. See the diagram: glycoproteins were the proteins with carbohydrate attached, glycolipids were the lipids with carbohydrate attached, and below you can see peripheral membrane proteins, integral membrane proteins, cytoskeletal filaments, cholesterol, protein channel and phospholipid bilayer. Next, functions. Main function of cell membrane: transport of molecules. Obviously, the membrane selectively permeable to some molecules present on either side of it. Due to the fluidic nature, the membrane can help in cell growth, formation of intercellular junctions, secretion, endocytosis, cell divisions, etc. Next is types of transport: passive transport, active transport. So see this. First of all, passive transport: movement of molecules across the membrane along the concentration gradient, that is higher concentration to the lower concentration, without the expenditure of energy, that is called passive transport. You can see the diagram. It is of two types: simple diffusion, which is the movement of neutral solutes across the membrane, and osmosis, movement of water by diffusion across the membrane. Here it is given polar molecules cannot pass through the non-polar lipid bilayer, so they require membrane carrier proteins for their transport, which facilitate the diffusion process. In active transport, movement of molecules across the membrane against the concentration gradient, that is from lower to the higher concentration, with the expenditure of energy or ATP utilized. As you can see in this diagram. Example, here sodium and potassium pump. Here the movement of molecules against the concentration gradient, that is called active transport, and here you can see ATP is utilized. Second is cell wall. It is a non-living rigid structure found outer to the plasma membrane of the fungus and plants. Cell wall of algae is made up of cellulose, galactans, mannans and minerals like CaCO3. In other plant, it consists of cellulose, hemicellulose, pectins and proteins. Next, cell wall of a young plant cell. Primary wall is capable of growth. It gradually diminishes as the cell matures, and the secondary wall is formed on the inner side towards the membrane. The middle lamella is a layer mainly formed of calcium pectate. It glued the different neighboring cells together. That's called middle lamella. Okay, you can see in this diagram middle lamella, primary cell wall, plasma membrane. The cell wall and middle lamella may be traversed by plasmodesmata which connect the cytoplasm of neighboring cell. You can see plasmodesmata also labeled in the diagram which connect the cytoplasm of the neighboring cells and they act like a channel between two plant cells. Function: it gives shape to the cell, it protects the cell from mechanical damage and infections, it helps in cell to cell interaction, it acts as a barrier to undesirable macromolecules. Macromolecules means large molecules. Third, endomembrane system. It is a group of membranous organelles having coordinated functions. They include Golgi complex, lysosomes, vacuoles. You can see in this diagram nucleus where the nuclear envelope is attached with the RER, and then you can see SER, smooth endoplasmic reticulum, and then after SER you can see Golgi apparatus is attached and where the lysosomes and vesicles are formed. First of all, ER. These are a network of tiny tubular structures scattered in the cytoplasm. ER divides the intracellular space into two compartments, luminal or inside ER and extraluminal, that is cytoplasm. ER is two types, rough and smooth. RER bear ribosome on their surface. RER is frequently observed in the cells actively involved in the protein synthesis and secretion. They are extensive and continuous with the outer membrane of nucleus. SER where the ribosomes are absent is the major site of synthesis of lipids in animal cell. Lipids like steroidal hormones are synthesized in SER. Golgi apparatus. It is densely stained reticular structure near nucleus, first observed by Camillo Golgi in 1898. They consist of flat, disc-shaped sac cisternae of 0.5 to 1 micrometer in diameter. These are stacked parallel to each other. Cisternae are concentrically arranged with convex cis, which is the forming phase, and concave trans, that is the mature phase. As you can see here, two phases are there. One is cis phase, it is labeled here, and trans phase is this side. It is the forming phase, or other is the mature or trans phase. Cis and trans phases are totally different but interconnected. They both are the totally differences but interconnected. Here you can see cisternae are also there, and anything from the ER and nucleus first enter through the cis phase and then it synthesizes between the cisternae and then it comes out the vesicle form. Okay, secretory vesicles. These are the newly formed vesicle, and when it detached from the Golgi body it is called secretory vesicle. Okay. Function: secretes materials to the intercellular targets or outside of the cell, materials to be packaged as vesicles from the ER fuse with the cis phase and moves towards the trans phase. As I told you, this is why Golgi apparatus remains in close association with the ER. Proteins synthesized by ribosomes on the ER are modified in the cisternae of the Golgi apparatus before they are released from its trans phase. Formation of glycoproteins and glycolipids done by Golgi apparatus. Next is your lysosomes. Membrane bound vesicular structure formed by the process of packaging in the Golgi apparatus. Lysosomal vesicles contain almost all type of hydrolytic enzymes, that is hydrolase, lipase, protease, carbohydrates. They are active at the acidic pH. They digest carbohydrates, proteins, lipids and nucleic acids inside the cell. Next is vacuole. These are membrane bound space found in the cytoplasm. It contains water, sap, excretory product and other materials which are not useful for the cell. Vacuole is bound by a single membrane that is called tonoplast. In plant cell, the vacuole can occupy up to 90% of the volume of the cell. Next, in plants, tonoplast facilitates the transport of ions and other materials against the concentration gradient into the vacuole. Hence, their concentration is higher in vacuole than in the cytoplasm. In amoeba, the contractile vacuole helps for excretion. In many cells, example protists, food vacuole are formed by engulfing the food particle. Next, your mitochondria. These are clearly visible only when stained or colored. Their number, shape and size per cell are variable depending on the physiological activity. It is sausage shaped or cylindrical, having a diameter of 0.2 to 1 micrometer. Averagely we can say 0.5 micrometer and length is up to 1 to 4.1 micrometer. Mitochondrion is a double membrane bound structure with outer membrane and inner membrane. As you can see, outer membrane, inner membrane. It divides lumen into two aqueous compartments, outer compartment and inner compartment, that is called matrix. The inner membrane forms a number of enfoldings that are called cristae, which are also labeled in here. Towards the matrix, they increase the surface area. Next, the two membranes have their own specific enzymes associated with the mitochondrial function. Matrix possesses a circular DNA, a few RNA molecules, ribosome 70S and compartments for protein synthesis. The mitochondria are divided by fission and the function of mitochondria are they are the site of aerobic respiration. They produce energy in the form of ATP. That's why they are called powerhouse of the cell. Next is plastids. These are found in all plant cells and in euglenoids. Euglena. Large sized, easily observable under the microscope. They contain some pigment. Based on the pigments, plastids are three types: chloroplast, chromoplast and leucoplast. Chloroplast contains chlorophyll and carotenoid pigments. They trap light energy for photosynthesis. Chromoplast contain fat soluble carotenoids pigment like keratin, xanthophylls, etc. This gives a yellow, orange or red color. Leucoplast, these are colorless plastids of varied shape and sizes with stored nutrient. They include amyloplast, which store the starch, example potato; elaioplast, which stores oil and fat; aleuroplast, which store proteins. You can see the diagrams. Chloroplast. These are double membrane bound organelles mainly found in the mesophyll cells of the leaves. They are lens shaped, oval, spherical, discoid or ribbon like organelles. Length is 5 to 10 micrometer and width is 2 to 4 micrometer. Their number varies from 1, which is present in the chlamydomonas green alga, or 20 to 40 per cell in the mesophyll. Inner membrane of chloroplast is less permeable. The space limited by the inner membrane of the chloroplast, that is called stroma, that is also labeled in the diagram. It contains a number of organized flattened membranous sac, that is called thylakoids. Membrane of thylakoids encloses a space called lumen. Chlorophyll pigment are present in the thylakoids. Chloroplast. Thylakoids are arranged in stalk called grana or the intergranal thylakoids. Grana or granum you can see in the diagram. They are flat membranous tubules called the stroma lamellae connect the thylakoids of the different grana. Stroma lamellae, which connect the two grana. Okay. The stroma contains small double-stranded circular DNA molecules, ribosomes and enzymes for the synthesis of carbohydrates and proteins. The ribosomes of the chloroplast are smaller, 70S, than the cytoplasmic ribosome, which are 80S. Next is ribosome. These are non-membranous granular structure composed of ribonucleic acid and proteins. George Palladay first observed the ribosome in 1953. Here you can see 3 ribosomes and ribosome associated to the RER. Eukaryotic ribosome has 2 subunits, 60S, which is a larger subunit, and 40S, which is a smaller subunit. They together form 80S. And the smaller subunit is made up of 16S rRNA and 30 proteins, while the large subunit is made up of 28S rRNA, 5.8S rRNA, 5S rRNA and 45 proteins. Objective question will be asked from these points. Next is cytoskeleton. It is a network of filamentous proteinaceous structure present in the cytoplasm. It provides mechanical support, motility, maintenance of the shape of the cell. Cytoplasm. Here you can see cell membrane and inside the organelles as well as the cytoskeleton, how it is present in a proper way, in a skeletal way, as you can see. Intermediate filament, microtubules, actin filaments. Next is cilia and flagella. They are hair-like outgrowths of the cell membrane. Cilia is a small structure which work like oars, causes the movement of cell or surrounding fluid. Flagella is the longer, responsible for the cell movement. Flagella of prokaryotes and eukaryotes are structurally different from each other. Cilium and flagellum are covered with plasma membrane. Their core, axoneme, has many microtubules running parallel to long axis. You can see microtubules are many are present here. Central microtubule, peripheral microtubule. Okay. Axonemes has 9 pair of doublets of radially arranged peripheral microtubules and a pair of central microtubule. This is called 9+2 array. The central tubules are connected by bridges and are enclosed by a central sheath. As you can see, it is connected to one of the tubules of each peripheral doublet by a radial spoke. Thus, there are 9 radial spokes are present. The peripheral doublet are also interconnected by linkers. Cilium and flagellum emerges from centriole-like structure called the basal bodies. Centrosome and centriole. Centrosome is an organelle contains 2 non-membrane bound cylindrical structure called centriole. They are surrounded by pericentriolar material. The centriole lie perpendicular to each other. They are made up of 9 evenly spaced peripheral fibrils of tubulin. Each of the peripheral fibril is a triplet. As you can see in this diagram, the adjacent triplets are also linked. So centriole, structurally it looks like in this diagram. The central part of the centriole is also proteinaceous and called the hub, which is connected with tubules of the peripheral triplet by radial spoke made up of proteins. The centriole form the basal body of the cilia or flagella and spindle fibers that gives rise to spindle apparatus during cell division in the animal cell. Next, your nucleus. It was first described by Robert Brown, 1831. The material of nucleus stained by the basic dye, which given the name chromatin by Fleming. Normally, a cell has only one nucleus. Some cells have more than one. Some mature cells lack of nucleus. Example: mammalian RBC and sieve tube cells of the vascular plant. The interphase nucleus contains nuclear envelope, nuclear matrix or nucleoplasm. See the diagram: chromatin and nucleolus. First of all, nucleus. Nuclear envelope, double layered membrane with a space between 10 to 50 nanometer called the perinuclear space. It is a barrier between the material in the nucleus and cytoplasm. Outer membrane usually continuous with the ER and also bears ribosome on it. Nuclear envelope has minute pore formed by the fusion of its two membranes. These are the passage of the movement of RNA and proteins between the nucleus and cytoplasm. Nuclear pore is also present, as you can see in this diagram. Nuclear pores which act as a channel between the nucleus and cytoplasm, which transport RNA and proteins. Chromatin, a network of nucleoprotein fibers called chromatin. It consists of DNA and basic proteins, histone, non-histone proteins and RNA. During cell division, chromatins condense to form chromosomes. Nucleolus, one or more non-membranous spherical bodies called nucleolus. It is continuous with the nucleoplasm. It is the site for the ribosomal RNA synthesis. Chromosomes. A human cell has about 2-meter long DNA distributed among its 46 or 23 pair chromosomes. Every chromosome has a primary constriction centromere, on the sides of which disc-shaped structure called kinetochores are present. Based on the position of centromere, chromosomes are of four types. Metacentric chromosome. Middle centromere formed two equal arms of the chromosome. The first one is the metacentric. Submetacentric, where centromere is nearer to one end, forming the shorter arm and one longer arm. Third, acrocentric chromosome. Centromere is close to its end, forming one very short and one very long arm. And telocentric chromosomes are the terminal centromere. Some chromosomes have non-staining secondary constrictions at a constant location that are called satellite. You can see the diagram, satellite also labeled. These are microbodies. These are membrane-bound minute vesicles that contains various enzymes present in both plant and animal cell. Microbodies. Difference between plant cell and animal cell. Plant cell wall present, animal cell absent. Plastids present in plant cell, absent in animal cell. Large central vacuole present in plant cell, which are many small vacuoles are present in animal cell. Centriole are absent in plant cell, while animal cell having centriole. And to comparison between prokaryotic and eukaryotic cell, here you can see prokaryotic cell generally smaller, eukaryotic cell larger. Genetic material is in the form of nucleoid, where the nucleus is present in the eukaryotic cell. Nuclear membrane is absent in prokaryotic cell but present in the eukaryotic cell. Membrane-bound organelles are absent in prokaryotic cell while it is present in the eukaryotic cell. Circular DNA in prokaryotic cell, linear DNA in eukaryotic cell. Ribosomes are 70S type while the eukaryotic cell having 80S type but 70S in plastids and your mitochondria. And with this, we have completed the whole chapter of cell, the unit of life of class 11th. I hope the diagrams and the points of your NCERT are helpful for the preparation of the school exams as well as the particular board exams. Okay, so watch this video. If you find this video helpful for you, then like this video, comment if you want, and you can share it with your friends, and do not forget to subscribe my channel. Thank you. Jai Jagannath.